tonight to go to New York. So it's really great that it's here. I think this talk will really provide great motivation for some of the tools that we're going to be using. So,
cool, thanks. Yeah, so the hope is that this will give you sort of context for all of the cool stuff that you want to go learn and do for the rest of the week. First thing to say is feel free to jump in with questions whenever I have material, but I don't even have to make it through all the slides. I'm going to do a little demo at the end of new platform we've been working on, but otherwise I want to make this as sort of interactive as possible, so don't hesitate to raise your hand. Does anybody know what this is? Phrenology. So phrenology, somebody want to tell us what phrenologists were, what they did? Yeah, Dan.
You looked at the shape of the skull and tell whether someone's immoral or
you tried to infer something about the psychological faculties, because they thought that if you're really moral, you're going to have a big bump in some particular place, and they could feel that in your skull. Now that's clearly, if you think much about the fact that brain is like jello and skull is really hard, it doesn't really make sense that your brain is going to be pushing your skull out. And so the problem with phrenology was, one, they were based on some bad assumptions, but more importantly, they didn't really sort of test hypotheses, right? They would basically say, oh, hey, you have a big morality bump, you must be very moral, right? So sort of like astrology. Clearly, we're doing much better with fMRI, right? We're asking questions in ways that are going to prevent ourselves from just generating bullshit in the way that things like phrenology or astrology or palm reading or, you name it. We think that those are primarily not ways to generate true knowledge about the world, whereas science is, right? We would hope. But nonetheless, in the last 10 years or so, we've seen a ton of evidence that there are fundamental problems with the way that science is being done. So, it's sort of first came out in psychology, but it's also been coming out in lots of other areas. And, in cancer biology, in lots of different parts of science, it's become clear that some of the ways that we do science are really problematic, leading us to potentially irreproducible results. Psychology, I think, has, even though some people view psychology as kind of like the crux of the problem, in some ways it's been brought on by psychologists themselves, because we've kind of, as a field, have jumped in and actually tackled the problem. So in the Reproducibility project, this group tried to replicate 100 published findings from a set of psychology journals, and basically found that they could, if replication is defined as getting a significant result, when the first project had gotten a significant result, they could only replicate about a third of them. Now, there's lots of examples from other domains. I like this one from John Ioannidis. Oh, actually, I didn't make it to putting in I was just doing this right before we were talking. I didn't make it to putting in the results. But basically, they went and looked across the literature for, can you find evidence for or against some particular ingredient causing cancer, or some particular food causing cancer, and basically found for a ton of different foods, you can find strongly significant evidence both that it causes cancer and that it prevents cancer. And the argument being that basically most of these are probably null, sort of irreproducible false positives. So Ioannidis wrote a paper a few years ago that, if you haven't read, I think is essential reading, Why Most Published Research Results Are False. And basically he makes the argument, and I'll go through the mathematical argument in a bit, but he makes an argument that there are sort of a set of things that drive fields towards being more likely to generate false positive findings. And those are things like the smaller the studies you do, the greater the number of statistical tests and the lesser of selection of those tests, the greater flexibility in the designs and the analyses that you can do, and the hotter the field with more scientific teams involved. So all those things can kind of lead you to think that it might be a problem for neuroimaging. This is just, we haven't had the same kind of project that psychology had in terms of trying to go and replicate a lot of different findings, but there is at least some evidence of big problems. So this came out earlier this year. It's kind of a re-meta-analysis of sort of revisiting a number of previous meta-analyses. These previous meta-analyses had looked at basically consistency of activation differences between depressed and healthy people on a set of tasks. And the earlier meta-analyses had basically said, hey, look, yeah, yeah, there's consistent activity in particular areas. This meta-analysis, which is done by sort of the best meta-analyzers in the world, basically came back and said, hey, when you fix the problems of those previous meta-analyses, there's no consistent differences in activation between depressed and healthy people on any of these different tasks, suggesting that that it's really easy to fool ourselves. So I like to think of neuroimaging as potentially a perfect storm for irreproducibility, and I think the goal of all of us being here, or at least of a lot of us being here, is to try to fix this, right? We don't want to be involved in an enterprise that's just generating bullshit. We want to generate truth about the world. So we started a center a couple years ago. This is work with Chris that is aiming at trying to fix these problems. We call it Center for Reproducible Neuroscience. Does anybody know what this is? If some of you have seen this talk before, so you can't answer. Yeah.
Is it a DNA sequencing machine?
Good one. No, actually, it does look like a sequencing machine, doesn't it? Any other guesses?
Sorry.
It's actually it's a commercial paper cutter. So you put in a big whole thing of paper in it, right? So you see it has two green buttons there. Why would a paper cutter have two green buttons? So that you don't stick your hand in like this and hit cut and cut your hand off. Right, exactly. So so the designers of this machine anticipated the stupid things that humans are likely to do and designed the machine to prevent them from doing that. And that's what good design does, right? It affords the things we want people to do and disaffords the things we don't want people to do. So we think about, one way to think about this is improving what's called the choice architecture of science. So behavioral economists talk about choice architecture as, when you go into a situation, any sort of human design situation, there are going to be some actions that are afforded and some that are disafforded, right? So when you walk into the grocery store, the things that are right in front of you, you're more likely to buy than the things that are hidden away on some back shelf. And so we can think about how do we design a scientific enterprise to make it more likely that people are going to do the right thing, less likely that they're going to do the bad thing. And that can involve incentives, it can involve nudges, like giving people the right defaults, providing the right feedback, and expecting them to make the kind of errors that we know they're going to make and trying to prevent those errors. So what are the things we have to worry about? It seems like one of the biggest things is the low power of neuroimaging studies. This is a previous study by Kate Button and colleagues from a few years ago that looked at sample sizes actually not in functional imaging studies. They looked at structural imaging studies and then in rat studies, and basically found that the modal level of power was down below 10%. So most studies are basically useless, right? If a study has power of less than 10%, which means that if there's a true effect, you have less than a 10% likelihood of actually finding it, that basically means that the study's futile, that you shouldn't even do it. Yet tons of those studies are being done. So when we look at the neuroimaging, the functional neuroimaging literature, this is based on data both from a manual meta-analysis that Sean David had done and also data from Tal's Neurosynth project. This is showing sample sizes on a log scale over the course of years. You see that it's gone up, right? And the median sample size in 2015 was 28.5. And so what you can do is you can kind of back out what's the effect size that you're powered to find. This is work that Joka Dernes did. And basically you're powered to find for a single 200 voxel activation with a power of about, you can find an effect size of 0.75 within that 200 voxel activation, right? So that may well be, 0.75, that's 0.75 standard deviations, right? In psychology or in sort of standard statistics, you think of that as a large effect size. And so the question is, what's a reasonable effect size for fMRI? It might well be that all of our effects are so big that this is a perfectly good sample size. So the problem is you can't just take the reported effect sizes and look at them because those are biased, right? Because they're generally going to be taken from a statistic that was found by searching across a bunch of voxels, and so they're going to have what's called the winner's curse. They're going to be bigger than the actual effect size. So we use the human connectome data to try to basically estimate unbiased effect sizes. So we took the regions that the Human Connectome Project had predicted would be activated in these tasks, and then we refined those areas using neurosynth. So for example, we'd say, if they predicted for the motor task, it would be pre-supplemental motor area. So we took that anatomical pre-supplemental motor area, and then we took neurosynth and said, find the voxels that are active for motor stuff. And that sort of helped us pare it down to try to find the voxels that are really likely to be activated for motor stuff, but in an independent way from this actual data set. These are the percent signal changes that you get within those regions, showing that a 4, for example, in the precentral gyrus, you get about a 3% supplementary motor cortex, you get about a 4% signal change in the motor task. That's about as big of an effect as you'd ever expect in an fMRI study, suggesting we are finding really active areas. But that's the average signal sort of within a person. The effect size that we really care about when we're doing a group analysis is what's the distribution across people? And that's what this Cohen's D tells us, is basically how wide is the distribution across people. These are the effect sizes in that same unit of standard deviations for all these different tasks. And what you see is basically the only one that even approaches that 0.75 is basically supplementary motor cortex activity when people are moving body parts. So that's like the biggest activation you would ever expect in an fMRI study is all your power defined. So for anything interesting, which is often going to be an order of magnitude weaker than those big finger movement things, you basically, it's futile to even do the study. Yeah.
Is that for, can I ask questions?
Yeah, please.
Were those all one sample t-tests?
Yes. Yeah, so it's just one sample t-test for activation versus 0. So you might say, well, we're underpowered, but that's fine, because that just means that if it's there, we won't find it, but it doesn't increase our type 1 error. And that's true. It doesn't increase your type 1 error, which is defined as the number of false positives over all tests, right? But what it does increase is the likelihood that when you find a significant positive result, that thing is false. This is what, it's the inverse of what Ioannidis and others call the positive predictive value. So the positive predictive value is basically, what's the likelihood of this thing being true when I say it's a significant effect, right? Because now you're conditionalizing not over all tests, but only over the positive outcomes. So think about it this way. Let's say you have 0 power. So you have no ability to find a true effect if it's there. But your false positive rate is 5%. So you're still going to have 5% of your tests coming out positive. That means that 100% of your positives are false, right? As your power goes up, the 5% is going to stay constant, but the number of true positives is going to go up, right? So your positive predictive value is going to go up. But that means that when you have really low power, most of the things that you find to be significant are going to be false, because they're all going to be false positives. You're also going to suffer more greatly from this winner's curse, from the idea that when you find an effect with low power, the size of that effect is overestimated. And Tal pointed this out nicely, 10 years ago now in his paper around circular analysis. So one thing that one often hears, sort of depressingly, is something like this: my result isn't significant, so I need to add more subjects. I'm going to come back and talk more about this Simmons paper in a bit, but that's really problematic, as they showed. And this is another paper that everybody should go read. It's called False Positive Psychology. That basically, if you, let's say that you, so this is this top line here is minimum sample size 10, and then collect. Basically, the idea is you collect 10 subjects, and then you start looking after every subject whether the result is significant, and then you stop when you find a significant result. If you do that, your false positive rate, which should be 0.05, turns out to be 22%. So you're much more likely to find a false positive. And that's because you're basically capitalizing on chance. You're capitalizing on the fact that, with small samples, things are going to bounce around a lot. So it's going to sort of, bounce down below 0.05, but you collect 2 more subjects, it might bounce back up. So how do we fix this? Well, one is you basically need to always determine the sample size before doing the study, unless you're going to use some really fancy methods that can sort of get around these issues that people like in clinical trials use. For almost everything that we do, basically you should be doing a power analysis, writing down what the sample size is. Often you can't do a power analysis because you don't have the data that need to go into it. Then what you should do is power for a reasonable effect size, which is not 0.75. It'll be a much smaller effect size. And there are tools to do that. So Jeanette wrote a tool a long time ago for doing power analysis on regions of interest. Yoka Durnez has a set of tools that you can find at neuropowertools.org that will do this given an image. And I'll show you, I think I'll actually, maybe I won't show you in a bit. But if you have data that are in the NeuroVault database, it's sort of really easy to click a button and actually generate a power analysis to say, how many subjects would I need to replicate this particular finding? I wrote a blog post a while ago called "Why Pre-Registration No Longer Makes Me Nervous," which is sort of a response to this famous psychologist who'd written a thing about why pre-registration makes her nervous. But so we're now pre-registering everything we do in the lab because it's become really clear that if you don't, then there's just way too much flexibility in how you do stuff. And so one thing to say about this, and this is probably something that we can come back and talk about, is almost everything that I'm going to tell you to go do is going to make your life harder, right? It's going to make it, now that we pre-register everything, we have a bunch of null effects, right? Because we can't go hack our way, even though we weren't explicitly, hacking our way to positive findings, you're much more likely to get a null result, because if you're doing interesting science, most of the things you're going to test are going to be wrong, right? If everything you're testing is right, then you're not doing very interesting science. And so having said that, if you want to end your career and be able to say, hey, look, I did a bunch of stuff that I can actually believe, then this, I think, is one of the most important ways to do that, is write down what you're going to do, sample size, analyses, hypotheses, before you ever do it, register it on somewhere like Open Science Framework or aspredicted.org, and then you'll have much more confidence in sort of what you're doing. And just want to point out, this doesn't mean that you can't go do other stuff with the data, right? You can pre-register a set of hypotheses. Things are going to change, and you can also go do exploratory analyses. The most important thing is when you write the paper, just be very clear. These were the hypotheses that were registered. We tested these. We also went and did a set of exploratory analyses, and the reader can then sort of, take for themselves how much they believe the pre-registered stuff versus the exploratory stuff. The most thing is just to be really clear. Yeah?
Something that came up in our group recently, did a pre-registered analysis, did the analysis, got results we completely weren't expecting.
Yep.
And so instead of publishing this paper that's got more than 90% exploratory analysis,
yep,
because we're trying to explain what the hell happened, we're going to pre-register our follow-up analysis. Is that valid?
If those follow-up analyses are based on looking at the same data set, then that seems like a problem.
Right. So yeah, we're going to get a bunch of additional data sets.
Yeah. No, that seems like a very reasonable thing to do. Yeah. Okay. So we've done that. I mean, we have a couple pre-registrations that have been sort of incremental, where we basically said, we're going to register this. And it was like one where we had a large data set where we said, we're going to take 200 of the subjects, they're going to be our discovery data set, we're going to hone our analyses on those and then apply them on the remaining 300. But we'll come back and pre-register those analyses before we touch those 300. Now you have to trust that do that. I mean, we all have to trust each other, right? I think that's fundamental. I mean, I'm not even going to worry about fraud here, because I think, that's just sort of, one, it's so far out in the tail that I think we can't do anything about it. And I think we just have to trust each other. But yeah, so that's the way I think about it, is sort of, it's totally fine to do it incrementally as long as there's no circularity such that the data that were used in those analyses are going to be tested again.
There are ways of actually taking some of the information and conditioning that information to proper inference on it. But there are like techniques.
Right.
Otherwise, you just have to split the data.
Right. So one thing that we've started doing for regimes where you can collect a lot of subjects is this kind of like discovery validation split, where we basically say we're going to collect a bunch of subjects, randomly assign them to discovery or validation, and then only analyze the discovery data set, torture that data set, and then come up with what we want to do that then gets applied in a registered way to the validation set.
How does it extend it to public data sets?
That's really challenging, yeah, because you can't.
A lot of people are using it.
Yeah, you can't unsee what you've seen, right? So I think we have certainly. So actually, this is a pre-registration of analyses of a public data set, of the Abide data set, and another data set from Open fMRI. So again, you have to trust us that we haven't gone and looked at the data sets already, or that nobody else has kind of shown us stuff that is relevant. But at least we wrote down what we were going to do, right?
The result of the data.
We've seen the results of the literature, yeah. So it's clearly less compelling than it would have been if we'd said we're going to go collect a new data set. But nonetheless, this was a machine learning classifier thing. We want to classify image quality of manual ratings. So at least we said these are the classifiers we're going to try. These are the criteria we're going to use, and so on. So I think I think part of the reason people are often scared off of pre-registration is that it can be viewed as kind of an absolutist thing. We see it as another tool, right? It's a tool to help us keep ourselves honest, so we use it in whatever way we can. So I think you don't have to be absolutist about it. Yes, Sandra?
I think just to add to the public data set point, a lot of public data sets that are coming online are prospective data sets, which will in fact lead you to do kind of doing the validation when the next 1,000 subjects are acquired. So you can do a lot of things with the current data that's released in this exploration phase, and then predict that the next collection of data is what you're going to use for validating the results.
Yeah, no, that's a good point.
I have a question. So when you say pre-register, does it mean to predefine your testing, validation, and training set?
What I mean very generally is predefine as many of the details about the study as you can. So certainly if it's an empirical study, predefine the sample size and the inclusion and exclusion criteria, and then predefine what's the analysis stream going to be in as much detail as you can. Obviously that can be hard. And then maybe most important, predefine what are the outcome measurements that you're going to use. So if you're doing, let's say you want to predict, who's going to end up in jail in a year based on their brain activity, right? And you're going to use a particular region of interest on a particular task. Write that down. Say, this is what we're going to use to predict, because otherwise there's just too much flexibility. You can always go find something that's going to predict, which is sort of the point made by this slide. Yeah, sorry.
So do you think this is complementary to the clinical trial registry? Because the crux with the clinical trial registries is they're often on the statistics side and power side very well defined, but on the analysis side, much less so. There's just, we're going to measure this with image analysis.
I think that we have a lot to learn from what people have done in the clinical trials domain. I think you're right that there are things that there have been problems in the clinical trials domain that haven't solved all the problems that people would hope clinical trial registration would solve. So I think we hopefully can learn from that and be more detailed about those things in these kind of pre-registrations.
One big problem with this thing that, I'm a big proponent, I like the pre-registration concept, but I feel like a lot of the people who use these big technological tools that for fMRI analysis specifically, they're from the clinical side. They're using them as tools that they learn through the process. And it's much harder to convince them to say, hey, learn all your tools first, pre-applying what you're doing, and then do it.
I agree. That's a place where the discovery validation set can be really handy, I think.
And I think that the biggest concern there is essentially that they don't know how to use these tools first off.
So another way to think about that, something we've been thinking about is the need for better data simulators. Because if I could give you a data set that looks exactly like the data set you're going to collect, and I know what the true signal is in it, you go find it, and then you come back, tell me when you're ready to touch the real data. I think that could be really useful. We don't have good tools right now for generating those data. So this is highlighting a problem with the fact that when we do image analysis, we have so many different voxels we're looking at. We're doing a ton of statistical tests, and if you don't control for them properly, you could end up in basically a land of false positives. So what I did was I created basically just a Gaussian random image for 28 sort of pseudo subjects, and then created a Gaussian random number that was this behavioral regressor, right? And I wanted to go in and find regions in the brain where there's a correlation between the behavioral regressor and activation, right? Where this activation is just random noise. And I used a thresholding approach that's not uncommon in the literature, like p less than 0.01, uncorrected, and then it has to be at least 10 voxels. And I didn't have to do this a bunch of times. I did it once, and I got this image, right? And there's a thing here that if I was studying, dyslexia, and I found this correlation in auditory cortex, I'd be like, wow, that's great, right? beautiful correlation between activation and behavior, right? That's just total noise. It's because we've, when you have 220,000 things, right, one of them is going to always look really good just by random chance. And you still see papers publishing correlations like this. Fortunately, less so than you used to, but you do still see them. Say that again? Something like that. I don't remember. Yeah.
So then, what's the suggestion? What would somebody who's doing this kind of threshold analysis?
The suggestion would be instead of using that thresholding method, to use a thresholding method that properly, I'll come to this, but that properly deals with multiple comparisons. So you may have heard of this thing that came out a few years ago, kind of a humorous example where Craig Bennett and colleagues put a dead salmon in a scanner and turned on the projector and showed it a task, like a theory of mind task, and then analyzed the data as if the dead salmon had been doing the theory of mind task, and saw, lo and behold, you get like a little blob of activation in the dead salmon's brain. If you don't do the proper correction for multiple comparisons. If you do the proper correction for multiple comparisons, you don't see that because it's noise. The field is over, and we may end up yelling at each other about this, we'll see. The field has for a long time been developing tools to try to address this problem, right? I think that people realized very early on, like in the 1990s in the PET literature, that we had to deal with this multiple comparisons problem. So there are both parametric approaches like Gaussian random field theory and non-parametric approaches like sort of randomization permutation testing. For many years, people have gone along using Gaussian random field theory in ways that turned out to potentially be problematic. And it particularly had to do with, so in Gaussian random field theory, what most people will do is use what we call cluster-based inference. The idea is you first kind of cut the map to find blobs, and then you look at the size of those blobs. And if you know the smoothness of the field, you can predict what's the distribution of the size of blobs you'd expect if there is no signal. And then you can put a p-value on sort of the size of the blobs. It turns out that if you don't cut the blobs high enough, the techniques perform badly. And in particular, they can have family-wise error rates that are highly inflated. They should be down here at 0.05, but in some cases they can reach almost 0.5. The question is, what do you want to do about this, right? And there's other problems probably with Gaussian random field theory that have to do with the degree to which the signal is actually Gaussian versus having long tails. And again, JB might disagree with this, but my inclination is to use non-parametric methods because we know that they're going to ultimately get us, as long as we're in the regime where we have enough data to do them right, they're going to get us as close as possible to the ground truth without having to make these assumptions about Gaussianity. So my lab has moved entirely to using randomization tests when we can. Obviously, there's some places where you can't, and then you have to think about other methods. But when we can, we use those. Fire away.
So it's sad because I don't entirely disagree. I wish I could disagree entirely.
Yeah.
But I think, just for the information of people, like the SPM threshold has never been 0.1. It's always been 10 to the minus 3. It's one of the zeros. In which case you get a false positive error of about maybe 90% or something like that, which is problematic, but way less problematic than all the other things that are happening in there.
Yeah.
That's one thing to say. The FSN threshold was actually quite a one.
Yeah. But they did other things that actually drove it in the other direction. Yeah.
But, so that's one thing. The other thing is that parametric methods are great, and when you use them, they are not always easy for a little bit of complex paradigms or a bit complex settings, and sometimes you actually do the wrong thing easily using parametric. So it's still a tool that gives you some visible result at some. If you're using it properly, it's still a visible tool that will give you something which is an approximation, and you should know that it's not exactly 0.05, it's maybe 0.07.
Right.
But it's still something that is very fast because it's analytic, and give you something which is
Totally fair. Yeah, yeah. So, and actually,
I think it depends on the setting. That sometimes you do need to do a lot of those tests, and neuroparametric is going to keep penguins again.
Right. We take advantage of really big supercomputers, so we don't have to worry about the time that it takes. But, so we'll actually come back to this p-value point in a bit. So another issue that we have is just how flexible our analyses are, right? So we put together this table for a paper that just went through, this is just in FSL, and figured out like, what are the number of possible things you could do at each step, and then just multiply those, right? So this is the number of possible workflows, 69,000. That's like about as many I don't even think there's 69,000 fMRI papers published, right? So you could have done a different workflow in each of them. Josh Karp did an analysis a few years ago looking at the effects of that, basically taking one of our data sets from OpenFMRI and running it through about 6,900 workflows, and showed that, in some places the range of the statistics is not very big, but in certain places this is the range of z-scores, right? So it could be in some places really huge. Suggesting that those choices could make a difference, right? Now, some of them are probably more reasonable than others. Nonetheless, it suggests that some of these things can make a big difference. So the issue of methodological flexibility has come to the fore in the context of what has come to be called p-hacking. Have people heard of p-hacking here? So the idea is that trying a bunch of stuff can sort of greatly inflate your false positive value. So this is I like this example from the Simmons et al. False positive psychology paper, where they did a study where they had people listen to undergrads listen to either this Beatles song or kalimba, and then they had them they recorded their age and their father's age, and then they used their father's age to control for variation in baseline age across participants. And Kovar revealed the predicted effect. According to their birth dates, people were nearly a year and a half younger after listening to the Beatles song than kalimba, right? So the p of 0.04, so it's significant. The point is that clearly listening to the song is not making people younger. They were able to capitalize on the fact that they did a ton of different analyses, which you can sort of see, like they did an ANCOVA with the controlling for variation in father's age. They tried a bunch of stuff until they found one particular combination of things that worked. And then they looked at, what happens if you combine, go back, you take two different variables that are correlated, you add 10 more observations per cell, control for gender or interaction with gender, dropping or not dropping one of three conditions. You do all those things at, p of less than 0.05, and your true false positive rate becomes about 60%. So clearly that's problematic. So one place that we feel like we might have seen this in the neuroimaging literature, so Jeanette, this is a little bit of background. Jeanette and I would have conversations for years about how we'd see papers where people would do all their analyses in SPM, but then they go get this AFNI tool called AlphaSim to actually do the control for multiple comparisons, right? And we thought it was a little bit cheesy, in part because we already knew that there were problems with AlphaSim that depended on how you specified the smoothness. And the AFNI people had given you ways to do it, but not everybody did it correctly. But we found, a decent number of papers when we looked through, this is like recent papers from 2015, that had actually done this thing where they analyzed with SPM or FSL, and then they went and used AFNI. It turned out that AFNI, there were a couple problems with AFNI. One, it had a bug that actually wasn't that big of a bug, but it did cause some problems. It also underestimated the smoothness, and that basically resulted in it having much lower cutoffs for activation compared to the other tools. So it was basically being overly generous. So it's as if people in the field were trying a bunch of stuff. They figured out, oh, hey, this AFNI thing gives us really good activation with our 18 subjects that were underpowered to find anything with, and they started using it, right? So that's a problem. So I've talked a lot about fMRI. It's not just fMRI that's a problem here. This is a recent paper from Steve Luck, who's like one of the huge players in the EEG world, where they basically did a similar sort of thing with ERP data, showing that basically if you have some flexibility about where you do your choices of cutoffs in the time series, you can basically get super high, like 50% false positive rates, just with basically noise data. So it's this is a problem, and it's not even a problem solely in neuroimaging. It's a problem that we think really sort of extends across any part of science that's using sort of large data sets with lots of methodological flexibility, which encompasses lots of what people in this building think about. I'm sure anything that involves quote unquote big data. So One of the things that we hope to ultimately do in the platform that I'm going to talk about in a bit, Open Neuro, is to be able to help people actually quantify how much, John Ioannidis has used this term vibration of effects, how much do your results bounce around when you try different workflows? And the point isn't to do 100 workflows and find the one that worked best. The point is to do 100 workflows and quantify the degree to which your particular results depend on the particular workflow as opposed to generalizing across workflows. So just how we think now about generalizing across people, we're going to think about generalizing across analysis workflows as well. So I like to, use the term that we should stop thinking of like hitting, finding a significant p less than 0.05 result as like hitting the jackpot. So many of you may have seen this paper that was bouncing around recently that basically argued that one thing that we need to do is to increase our level of statistical stringency. So we're talking about 0.05. They basically push to move it to 0.005, and the argument is that if there's sort of a one-to-one relationship between the p-value and the amount of evidence in favor of the alternative hypothesis versus the null hypothesis, right? So we'd like to be pretty sure that when we say there's a significant result, that we have a lot of evidence that there's a true effect and that it's not null. It turns out that when you look at 0.05, that amount of evidence is so they use this thing called Bayes factor, which is basically like the relative strength of the alternative versus the null. A Bayes factor of 3, which is what p of 0.05 roughly comes out to under a range of assumptions, is pretty weak evidence. Like most people wouldn't think that if the alternative is 3 times more likely than the null, that's not really great evidence. Whereas 0.005 comes out to roughly about 20 times more evidence for the alternative versus the null. Yeah?
Why not use Bayes factors instead of p-values?
Yeah. My answer, so I think Bayes factors are great, right? The problem is that they rely on priors, and you can actually push Bayes factors around, not greatly, but you can push, in certain cases, you can push them around quite a bit depending on the prior you choose. I mean, I'm a big fan of Bayesian statistics, but we don't yet have all the tools we need in neuroimaging to do them properly. I think if we did, I would love to be able to just compute, Bayes factor maps or posterior probability maps or something like that. But we just don't have the tools right now. Hopefully those will materialize. But I mean, this is not a debate. Many of you are probably familiar with these debates between Bayesians and frequentists that go back 50 years or more. And so I agree that that's one particular alternative. This paper discusses all of these issues. The paper is actually a nice, an interesting place where kind of frequentist and Bayesian stuff can come together in sort of showing that at least under some constraints they kind of map onto the same question. Okay, so turning to another aspect of reproducibility. How many of you have written computer code before? Okay, I figured most people in this room have. How many of you are trained in software engineering? Okay, actually a good number of hands. How many of you have ever written a test for your code? Wow, okay, I'm gonna have to change this. There's too many hands. So it's clear that one of the potential problems, I mean, we saw a minor version of this with the AFNI bug, but you can also see major versions of it as software errors. So Jeffrey Chang is a structural biologist. He had 3 papers in Science around 2001 to 2005 about the protein structure of this particular thing called the ABC transporter. All of which had to be retracted because of a bug, a single sign flip in the analysis that he was doing that totally changed the protein structure. Somebody else figured it out, and they sort of forthrightly went in and dug into what was going on. They had this piece of in-house software. Turned out it had a bug in it, and had to retract 3 science papers, not what one wants to do. So I wrote a blog post about a similar sort of thing that happened to me. I was doing an analysis of the open fMRI data set. We're trying to do basically, given an activation map, can we classify what task you were doing? And it was a decent sized data set, and we were trying to classify like 1 out of 27 or 28 different tasks. So chance is pretty low there, right? It's like guessing 1 out of 27. So I had a large number of subjects, like 300 something subjects, and I run my classifier. Pretty simple little bit of code like this, right, from using scikit-learn in Python, and I get 93% accuracy. Now, if I had gotten 100% accuracy, I would have been certain that something was wrong. But 93 was like far enough down from 100 that it wasn't clearly wrong. And so I was like, wow, look how well we can do at this. And then Sanmi Koyejo, who's a machine learning guy who was in the lab at that point, I sent him the code because he's a co-author on the paper, and he sort of looked at it and he's like, oh, you made an error. You forgot these 2 characters in your reference, so it was basically flipping the data the wrong way, classifying on the wrong thing, and now we're down at 53% accuracy. Still well above chance, right? It's not a bad outcome, but much less, thank God we didn't write the paper and report the 93%. The thing is, nobody would have ever found it, probably, unless some, grad student grabbed the code off my GitHub and started digging into it. So this points to a couple things. Is one, code review is a really useful thing, right? That it's always good to have somebody else looking over your shoulder. And two, that that tiny errors can have big effects. So I don't know if it was that same blog post, I've written a blog post about what I call the principle of assumed error, which is basically whenever something works out well in an analysis you're doing, you should assume that you did something wrong, that there's a bug that got you there. And part of the problem is that there's this asymmetry in what happens with bugs, right? If there's a bug that causes things to go against your assumptions or to disconfirm your hypothesis, you're likely to go in and find that bug, right? If a bug causes things to work well, not necessarily too well, but kind of well enough, you're less likely to go dig in and find that bug. So I like to refer to that as bug hacking, right? The idea that that sort of certain bugs are going to be less likely to be found than others, and always assuming that something is wrong sort of helps you get around that. And software testing, which you're going to hear more about, is obviously one way to try to get at that. You'll hear some examples of testing, and part of the problem is that the testing that people in the software world do isn't always the kind of testing that we need to do, right? You can write little unit tests for all of your individual functions, but that's not necessarily going to find the kind of problems you want to find. So the kind of things that we often do are, one, basically generate data for which you know the right answer. That are otherwise kind of reasonable, that look like the data you're going to be analyzing, and make sure your code can give you back the right answer. And it should be able to give you back the right answer both when that answer is null and when that answer is positive, right? And often what you'd like to do is kind of do a parameter sweep and make sure that it can actually give you, if you're estimating a parameter from the data, make sure that you can actually recover that parameter. We found some examples. For example, a psych review paper had a behavioral model that worked well in one little domain. At least in the paper, we got the model code, could not recover any parameters from like reasonably simulated data. And so randomization is another version of this. Basically, give it null data and make sure that it gives you back null. And one thing that's important is to make sure that you're replicating the entire analysis stream. Because if you have some sort of like circularity within the stream, you might not see it. So what you want to do is sort of treat your fake data as if they're coming from the same machine that your real data comes from, and do the entire pathway, the entire workflow. Okay. Yeah.
What kind of incentive structures do you think are going to come down the pipeline for people to discover bugs in each other's code? Like, the bug hacking thing applies to your own code, but it also applies to us sussing out bugs in other people's code, right?
Yep.
Like if someone's that prediction accuracy is way too high, there's got to be something wrong, I'm going to dig into it, right? That's what drives people to figure each other's problems, right?
Yep.
Do you think there's like something coming?
It's interesting that there's all this technical stuff that people are going to learn about here this week. That's not at all the hard stuff. The hard stuff is exactly this kind of social incentive structure type stuff that you're bringing up. If we don't fix that, it's going to be really hard to fix the rest of the stuff, because people who do good science are going to get out-published and outperformed by people who do crappy science. And so we're thinking a lot about that. So now when we this doesn't get back to code review, but when we look at people for we look at job candidates, we get to a short list. Obviously, we can't go in and read every paper of the 150 people who apply, but when we get down to a short list of 15, we go look at those papers and say, hey, this person looks like they do a lot of p-hacking. Maybe we're going to push them to the side, right? I think that's one of the kind of things we can start doing, but it's really hard to know how you incentivize this without the problem is you want it to be kind of a virtuous circle, not a vicious circle, right? And you can imagine it, in some ways all the reproducibility stuff has become a bit of a vicious circle where you have websites like Retraction Watch, right, where if you retract your paper, they're immediately almost like implying that you've engaged in fraud or something like that, right? When really we should all be, if I find a problem in a paper that I published, I want it to be retracted because I don't want it to be out there in the world, and that's how good science should work, right? So I think that there's a lot of social stuff we have to work on, and I don't feel right now. It's hard to feel super optimistic, at least in the short term, that that's going to fix itself. I hope that the fact that so many people of the generations that are sitting here are really enthusiastic about making this happen, that it sort of has to happen, but it's going to be a struggle for the next decade, I would imagine, at least. Yeah.
Maybe as a comment to this, I think the more tools you make available, especially also for the clinicians that at least in psychology, in neuroimaging, do also a bunch of the analysis, the more tools we make available for those people to use, so they don't use, oh, I follow what has been published 10 years ago, but they actually use our tool. I think that makes a big difference. And then I also think, when you review papers, look at the statistics. If it's bogus, then point it out.
And ask for the code to be shared if it isn't shared. And actually have a look at it. No, it's interesting. I just, so some journals are now, like Psychological Science now requires you to either, provide the data or say why you aren't. And I just recently reviewed a paper for them where I went in and looked at the data and something was clearly wrong with it. Like all the activation was in the ventricles. And they were talking about a bit that wasn't. I was like, look, this is bogus, and I think otherwise they would have published it and it would have been seen as fact. So I think it's really important to get both data and code as part of the review process to help catch those sort of things. Let's see, JB had a comment.
I was just mentioning.
Okay.
I think you mentioned that there are initiatives to actually review code quickly. I think as reviewers, we should be starting to really ask for the code. And the data, of course.
Right. Let's see, Chris H. had a comment.
Yeah, well, it's actually just more of a statement. Like, I think these are really interesting questions, and that figuring out those sorts of problems are going to be, it's going to require like a lot of critical thinking. At these sorts of meetings. So if that's like something that people are interested in talking about, how do you solve like not only the technical but also the cultural challenges there, it would be great for a conversation.
There's a phrase that I feel like I depressingly use more and more often, in part because it more increasingly applies to me, which is science progresses one funeral at a time. So part of this is just about regime change, right? It's about basically getting rid of the people who are now running things who grew up in the world where all of this stuff was fine, and having a generation of people who are attuned, who grew up, in the world where people care about reproducibility. I think, unfortunately, that's part of what it's going to take. Chris G?
Any advice on how to report such problems that we find only because someone shared the data?
Yeah, obviously I haven't always been so good about that. I think what we actually probably should think about doing is coming up with a sort of a set of best practices about how to actually comment on shared data. That probably involves, starting with the investigators and, surfacing your issues with them before you write about it in your blog, like I have in the past.
A good way to go about that would be to bring out like a best practices or instructional manuals for people who are not familiar with this kind of an issue. When they do data analysis, they can reference that manual and say, okay, I did this, I did do this, I did this, I don't do this.
Right. Yeah, that's. So we're starting to have some of that. So the OHBM just recently published this thing called the COBIDAS report, Committee on Best Practices and Data Analysis and Standards, and that actually has, for some of this stuff, actually has a lot of information about best practices for workflows and stuff like that.
So I'm talking about like an expanded version of the COBIDAS report.
Yes, right. Around kind of how to deal with issues that arise in analyzing shared data. I agree. I mean, JB and I wrote a little bit about this a while ago. We have a paper in TICS a few years ago called Publication Challenges of Shared Data that kind of addresses some of these points, but I agree that thinking about a set of kind of best practices there would probably be a good thing.
The key aspect is that we have to have some influence on the publication. That's hard, because publication usually must go back companies that we
Yeah.
Just kind of following up on that publication thing. I think that when I get a paper review, I think like most people doing kind of in the midst of everything else, and it's something that I feel is important, but like I want to do a good job and also not spend too much time on it. And I feel like it's important to review code, but it's something that might be interesting if some other people are interested in about this. Maybe like standards that journals might implement in terms of commenting code organization.
Yeah.
Reviewers can actually
Yeah, that's a really challenging one. I mean, in some sense, I agree. In another sense, that just becomes a bigger barrier to people actually being willing to share their code, right? So it's a real challenge there. I mean, obviously, we want everybody to be a better coder, and so I agree in that sense. But I don't want to tell people basically, you can't share your code until it is, well commented and has, 90% test coverage, when most people aren't going to get there. Right? Yeah.
At the risk of perhaps incentivizing more of a vicious cycle rather than a virtuous cycle, do you think that there's any space in this conversation for offering carrots to people for investigating and pursuing virtue? Like, I have a friend who's a postdoc, and he found a big problem in someone's code that basically led to pretty dramatically inflated correlations. And it ended up in something like kind of like a flame war between him and this lab.
Right.
He was really doing the right thing, but it seemed like it was just a lose-lose situation.
Yeah.
There was really no reason that he should do this besides his own, moral and ethical code.
Yeah. I wish I had a better answer for that. It's really hard for me to know what to say other than, it sort of points back to the fact that, people are viewing, many people in many labs view positive scientific results as hitting the jackpot rather than as a search for things that are actually true and generalizable and reproducible, right? And so I think that's the fundamental thing that we need to change, and that's hard, right? Because so many people have sort of come into science thinking that that's what you do in a lot of labs, right? So I think there's a deeper cultural change that has to happen around that. You would like to, it may well be that, as humans, there's no way to fix it, right? Because we always, we're always going to want to own ideas, right? Understanding how to get around sort of basic human foibles, and designing, systems that can allow us to have the virtuous rather than the vicious cycle, I think is a real challenge that I'm not prepared to figure out, right?
Maybe pushing for the non-static publication.
I see. I don't think that doesn't fix the.
It's going to solve it, right? But it's at least it would be like more common to see updates on something.
Yeah. Right. So if
more common will make it less dramatic in some ways.
Right. If papers could have versions and revisions and yeah, I agree that could start to at least get people to. Yeah, Grace.
Recently, I think it was at Ohio State. I saw that I think the paper was on if you're a good marksman in Call of Duty, you're a good marksman in real life. And they found that it was incorrect. And so the senior author who is at Ohio State, who I think at the time when they published it was a graduate student, could be a little wrong, she got fired, but the senior author on the paper didn't. And so I think another issue is at the university level. I mean, mistakes are going to, I don't know if it was on purpose.
Yeah. I think that I don't remember all of the details. Somebody else may remember better than I do, but there was a reasonable evidence of fraud in that case. I think it wasn't just somebody made a mistake in their code.
But I think what it comes to is when you don't know that it was fraud or not, it instills this sense of fear. Especially
no, I understand. Yeah.
The younger one got fired, but not the senior. So I feel like a lot of the carrots go to the lab or to the senior, and a lot of the ruler against the knuckles is on the grad student younger. If there's any paradigm shift within lab structures for that, if you had a comment.
Don't go to work for an asshole. That would be. No, seriously. I mean, I don't know how else you fix that.
Every guy is going to be an asshole, right?
It's hard to know. Yes, right. Yeah.
There are a lot of nice people who aren't as methods rigorous, and I feel like almost a lot of people, like being polite and being nice is like negatively correlated with being mean enough to be like, that's wrong.
So the point I was trying to make is that, you know, if you don't want to go to work for somebody who's going to throw you under the bus as a grad student so that they don't get, you know, attacked, right?
The point I'm trying to make is until the situation comes up, you won't know how the eyes are.
Well, sometimes you do. I mean, there are, you know.
But it's harder to.
No, especially when you're not already working in a lab and you don't have a social network. You can't ask around about somebody who's, you know, like legendarily abusive or something like that.
It's a matter of fact that as a grad student or a postdoc, you have less options in terms of what you're looking for compared to when you're looking for a job. So.
Indeed. Yeah. Yeah, no, I think I don't downplay these challenges. Just they're really hard because of the asymmetric information, because of the asymmetric power. It's really hard to know like how to fix those, other than trying to reward people who treat people in their lab well, and unfortunately that doesn't happen always.
I think though, like the OHPM initiative and the Open Science and the COVID-19 initiatives have kind of highlighted who are the people for these kind of initiatives, and people who associate with these people by association get.
I hope that's a valid signal.
Yeah, I mean, I think it's a valid signal. I think like for people who are looking for grad positions or postdoc positions, would think,
right. Yeah, at least you know that that's a lab that says it's committed to reproducibility. Yeah. Sorry, did you have a comment?
Yeah, I just wanted to. We talked about the higher stake case quite a bit, and I work with 3 different PhD students, and I work as much as I can closely with the code, but I mean, I cannot imagine people like JB or Russ, if you have that many students, there's a limit of what you can actually look at their code. So I think actually teaching them this stuff is the best way, teaching them the good practices. Otherwise, I really would think that my boss, for example, who is a clinician, she has no way of understanding like zero.
Yeah, no, I think that's a good point. Sorry, a comment over here?
Do you think the power structure can be normalized by putting pressure for open reviews, because it's mostly senior people who...
Can we not go there, please? Sorry, JB and I had an hour-long public argument 2 weeks ago about open reviews. Maybe not an hour long. I think that's like a discussion that should be had over drinks, I think. Just because I don't think it solves the problem. Other people do. I'll just leave it at that. I'm happy to come back and talk about it, but it's a bit off track here. So, okay, so let's talk about data sharing. So I, for a long time, have thought that one of the ways to try to fix this is to get people to share their data, in part because, again, it's a signaling process, right? It says, I'm comfortable enough to give you everything. If you want to look under the hood, you can. And also because it has scientific benefits, right? I actually got into it not so much because I'm a do-gooder, but because I wanted more data than my lab could collect to do these kind of large-scale analyses, and we thought that this was the way to do it. So obviously there's a trade-off in data sharing in terms of what you're going to share and how hard it is to do that. Sharing complete raw data sets is really hard, so of course that's the thing we started with. Tal started with the easy thing. Where is Tal? He started with Neurosynth, which is just like pulling stuff out of papers and putting it on a website. That's easy. It's actually not, but and then Chris is sitting here in the middle with NeuroVault, which is also really easy, as you'll see. Yeah.
Just got one comment on that, the structure there. The issues with code that are lower down on that pyramid wind up causing kind of more grief consequently, I think, because they are more potentially used. I got a little burned recently by like HCP 70 preprocessed data. Having a bug was another scientific thing. I have to like install a bunch of analyses that I was working on. So, maybe just be cautious with like processed data.
Indeed, yeah. You and I think having faith in those, knowing what the validation was that went into those processing steps is really important, I think. But you're right. I mean, there's almost certainly shared data out there that has errors in it. I'm sure that some of the data that we're sharing has errors in it, right? Because we're humans and everything we do is going to have errors. And the important thing is kind of minimizing those and figuring out how we find them and then reacting to them in the right way. But yeah, that was a really good example. I mean, I think they reacted in the right way, right? They didn't try to cover it up. They basically blasted an announcement that said, hey, there's a problem, hold on, right? But yeah, I agree. It's the more trust you have, the more faith you have to have. So we started sharing complete raw data sets 8 years ago or so now in the Open fMRI project. It's now up to like almost 3,000 subjects. And one of the things that we did that early on in the Open fMRI project was basically to say all the data in this project are going to be organized in exactly the same way, with the goal of being that I can give you a data set and we never have to have a conversation. You can take that data set and go analyze it, and you'll have all the information you need in the data set, and it's organized in a way that you can just look at it and find what you need based on the spec of our standard. So we had kind of come up with an in-house standard a few years ago, and then it became clear that it was sort of limited, and especially as we started thinking about going towards other types of data, like structural data and diffusion data. So we worked, Chris basically brought together a bunch of people, and not just because writing papers with 28 authors is fun. Basically, we wanted to get the consensus of the community around how should we structure a data set. So we came up with this thing called BIDS, Brain Imaging Data Structure. It's basically a way to organize a data set. It's not a file format, right? We rely on standard file formats like NIFTY or tab delimited text or JSON. It's really a way to sort of lay out the data. So for example, it tells you, what should the directory structures look like? What should the file names look like? And probably most importantly, what should the metadata look like? How should you describe, what was done in each scan, who the subjects were, all that sort of stuff. So if you go to the BIDS page, you can find a ton of information. And importantly, there's a validator. So if you have a data set and you want to know, is it a valid BIDS data set, you can just go to this website, point it to the data on your disk, and it doesn't have to upload the data at all. It's JavaScript. So it looks on your disk, tells you, are there any problems with your data set where it fails to properly follow the standard. You can fix those. And then once it's fixed, there's some cool stuff you can do with it that I'll come back to in a bit. I'll just mention NeuroVault is sharing raw data sets is hard, mostly because organizing big data sets is hard. People have thought for a long time that sort of sharing the statistical maps might be, the results from the study might be an easier way to get people sort of going with data sharing. Chris built NeuroVault to let people do this, and it has grown really quickly, has lots of data. There's now like a one-button export from SPM, and there's a script you can use to export data from FSL as well into NeuroVault. And so put your data in NeuroVault, and then you can just, actually I don't have images, but you can just sort of bring them up, poke around, somebody else can download them if you've made them publicly available. And there's also a bunch of tools you can get, like comparing it to the data in NeuroSynth and telling you what are the most common terms for images that look like yours, finding the most similar maps in NeuroVault, doing gene expression decoding, doing power analysis. There's lots of, the idea is to sort of incentivize people to share their data by giving them cool tools that they can access if the data are in the database. So why share data, right? One reason is that people who should, obviously, this doesn't mean that if you start sharing data, it'll cause you to do better science, but it is empirically the case that people who share data do better science. So this group basically asked a bunch of psychologists to share data from a particular set of psychological journals. They heard back from, I think, a third of the people. And then they have this tool called StatCheck that can go in and find errors in the statistics in the paper, like where the degrees of freedom and the p-value and the t-value don't match, stuff like that. And they basically found that people who were unwilling to share their data had more statistical errors in their paper than the people who were willing to share their data, suggesting that there is some relationship. Again, we don't know how the causality works. How many of you heard about this research parasites thing? Okay, we've already heard a reference to it this morning. So there are clearly a lot of people in the world who don't believe that data sharing is a good thing. These guys are, I think they're editors of the New England Journal of Medicine, at least one of them is. They wrote a thing, basically, there's some new guidelines coming on about sharing of clinical trials data, and they basically wrote this editorial griping about it, saying, in particular, there's some concern among some frontline researchers, i.e. actual scientists, that the system will be taken over by what some researchers have characterized as research parasites. So, you know, those of us who take data from HCP or ADNI or whatever and analyze it, we're parasites. And somebody came up with a great badge and a Twitter page, the research parasite. And there was this like huge Twitter storm about it, and they ended up writing another editorial, basically. We want to clarify. We really think that it's good. So, you know, some have spoken pejoratively in describing data scientists who apply data of others, but it's important to acknowledge and air those concerns. But we think that they can, you know, improve public health. So they kind of backed down a bit on their comments. But nonetheless, it's clear that especially in the medical world, there's a lot of concern, and some of it's well placed, probably, right? Clearly, you know, you want people to get credit for all the work that they've done. If you've collected this incredibly rare data set, and I'm telling you, hey, you have to put that online now and let anybody in the world analyze it, unless we can give you proper credit for all the work you did, that's not really fair in a sense. I would say that, you know, for people in our world, the biggest concern about sharing data is getting scooped, right? Having somebody else go do the thing you were willing to do or you wanted to do. There are very few actual stories of people getting scooped. I know one where someone claims they got scooped, but the data had been online for like years or for a long time, and it wasn't a completely, you know, non-obvious thing that was done. So I think that there's a lot more concern about getting scooped than there should be. So Gary King's a political scientist who has this great quote. Actually, what's much more important to worry about is getting ignored. Right? If nobody knows who you are, then it doesn't really matter if you're getting scooped or not, because you're not going to get jobs, you're not going to get grants. Right? What's really important is that people know about the work that you're doing and you know who you are. There's concern. We've already heard about, you know, concern about errors, and I agree that the way that the field deals with errors that have been discovered needs some work. But, you know, I think we should all want to know if something was wrong with what we published, right? We shouldn't want to hide any problems that we find. We should want to surface those, because that's what science is about, is finding the problems and fixing them. And then there's really legitimate concerns about, you know, the time and effort that it takes. Now, you already heard about the BIDS format. If you're sort of starting from the beginning in organizing your data using the BIDS format, and some of the databases are starting to work towards sort of like XNAT or whatever, generating your data in kind of BIDS format from the beginning. That makes sharing much easier down the road, both sharing with others, but also, like, you know, part of the reason I started doing this in terms of like organizational structure of data is, you know, I had some postdocs leave the lab, and then I wanted to go back and look at their data, and I had no idea what was what, right? Because they had organized it in some idiosyncratic way. So, so it helps you actually know, even I going back and looking at my own data, if it follows that structure, I know what I did. And then NeuroVault actually makes it much easier. So we've talked about sharing of data, we've talked about sharing of code. There's another issue, which is kind of sharing of bigger platforms. So David Donahoe, who's been a big reproducible analysis person for a long time, said that an article about a computational result is advertising, not scholarship. The real scholarship is in the code, the data, and the platform, right? Or the environment that produced the results. So many of you may have heard of My Connectome, which is a project where I scanned myself for a year and a half, and I had this huge data set, and if we sort of boiled it down from, you know, a bunch of imaging data to kind of time series that I could analyze on my Mac, right? So I was doing all these time series analyses in R, and I had already built a reproducible pipeline where I, you know, so for example, you know, several times the data, the pre-processing changed a bit from my colleagues at WashU. They sent me new data, and I had to be able to basically rerun everything. So I built a completely reproducible workflow, and had actually written the paper, put the results from that workflow in, but I wanted to make sure that it could actually run on another machine. So I copied it over to our Linux box, you know, took a couple hours to get all the dependencies going, and then I punched the button. It ran all the way through. And it gave me results that were different from the results I'd gotten from exactly the same code on my Mac. And it turned out that there were differences, even with the same version of a particular R package, there were differences in how it worked on Linux versus Mac or Windows, because I also sent it to somebody with Windows to try it out. They got the same results I'd gotten on my Mac. And so that got me really worried about even if I am doing everything that I've told you I should be doing, I could still, put something out and somebody could come and say, hey, I got different results than you on the same code and the same data. So that pushed us towards thinking about sort of software engineering tools for reproducible science. And there's a couple of different approaches. You're going to hear a lot about containers and Docker, and I think those are really good approaches. We started with virtual machines, which is sort of like me handing you a little computer in a computer. And this tools like Vagrant make that relatively easy, where I don't have to give you an entire 9 GB image of my virtual machine. I can give you just a file that describes it and tells it where to go get the code. It's not perfect if you don't wire in all of the exact software versions. I just had this happen recently where the R packages, I hadn't wired in the exact versions, and somebody wrote me and they said, hey, I did your gene expression analyses and you got 43 modules, but I'm getting 45. What's going on? It turned out that he had a newer version of the package that slightly changed what it does. So it's not, none of this stuff is, perfect, but nonetheless it helps us, be able to give somebody code and data with reasonable confidence that, modulo really weird stuff, they're going to get the same answer. Again, you're going to hear a lot more about containers. We're now using containers for a ton of the work that we do in the lab, especially for anything that we want to distribute, particularly around this project called BidsApp. So one of the ways that we're trying to incentivize people to use BIDS is basically to say, if your data are organized in the BIDS structure, you can just take these apps and immediately go run things like FreeSurfer or Tracula or Nutmeg or whatever, like code that would be reasonably complex to install. Now all you have to do is like one Docker command, and it'll just run, and it'll know, where all the subjects are in your data set that have T1 weighted images, and it'll just go pull them in. And so we now have a growing set of these apps. We just had a code sprint some of you were at last week at Stanford to sort of flesh this out and develop more of it. And part of what we're doing, I'm going to show you in a bit, give you a hopefully not a crash and burn demo of OpenNeuro, which is a new platform we're developing. The idea being that it takes in BIDS data, it lets you apply BIDS apps workflows to those data, and then lets you ultimately share the data and the results from the data, either privately or publicly. So part of what we've done is developed a set of these sort of BIDS apps around this project that we're starting to call the OpenNeuro Suite. We're basically, the problem with any of this stuff is you can get, the more powerful tool you give a person, the more likely it is that they're just going to apply it without knowing what they're doing, right? And without sort of checking to make sure that it's actually working correctly, right? So we have what we call a glass box philosophy, which is we don't want to give you a black box that just does stuff and gives you back data. We want to give you something that lets you look inside and particularly gives you reports on sort of how everything worked, so that you can go in and make sure that everything worked properly. So we want to be as reproducible as possible, so we use containers that are versioned, so you know exactly what version of FreeSurfer you ran on the data. You're going to hear more about testing and continuous integration from Chris. And then we're trying to be community driven to actually do stuff that people out in the world want, not just the stuff we want to do, though obviously we do the stuff we want to do. So a couple of the things we developed are Mri Qc, which is a fairly robust QC toolbox for right now just for structural and functional data, hopefully at some point for diffusion as well. That runs on our platform, but you can also just run it using Docker or Singularity on your own machine. We have another one called fMRI Prep that does fMRI pre-processing, including spatial normalization. Does it work on the surface now?
Yeah.
Okay, so surface and volume-based spatial registration, all the other good pre-processing stuff, field map, distortion correction, and it's robust in the sense that it looks at your data and figures out, oh, you don't have field maps, we're going to skip that, and so on, or we're going to do something that doesn't require field maps to fix the distortion, and it gives you reports. I'll show you some of these in a moment. So let's see, how are we on time? Okay, we're good. So I want to end with, I don't know how many of you are familiar with this paper. There's a set of these papers in PLOS Computational Biology about 10 simple rules that are all often really great papers. This one, 10 Simple Rules for Reproducible Computational Research, is really good and relevant to what you're all doing. I'm going to boil it down to 4 simple rules. The first being basically for everything that you're going to put in the paper, every result or every figure, keep track of exactly how it was produced. And that sounds trivial, but actually, the way many of us do science is not how that's done. And so a corollary of that is avoid and I think I'm preaching to the choir here, but avoid manual data manipulation if at all possible. So just get Excel, other than for like opening files to make sure that they're fine. Just don't use Excel to do anything with your hands. This is a great example of that. Have any of you heard of this? This fiasco. So this is a paper that was published by a couple of economists that is actually, it basically was the basis for all these austerity programs in Europe, because it sort of argued that more debt reduces growth. Reduces economic growth. So basically telling countries like Greece, basically you can't go into debt, right? It turned out that they had an error in their spreadsheet where like the formula was left out a few lines and basically left out the key counterexample to their claim, which was Belgium, right? And so they, somebody else, fortunately they shared the spreadsheet. Somebody else, later went and looked at it and realized, hey, you guys goofed up. All these like miserable people in Greece may not have needed to be miserable after all. And so obviously, that could happen with a Python script as well. We'll come to that in a bit. But nonetheless, it's it's probably it's possibly less likely to happen if you're writing code than if you're like pulling and stretching boxes. Okay, I don't think I have to preach to anybody here about version control, but when I started doing stuff, I would often have files like this on my machine, and many of you may have seen files like this as well. So GitHub or similar things help fix that nicely. So you're going to hear more about quality control and software testing. This is another great example of where like a little bit of testing could have gone a long way. So they, Lewandowski and colleagues wrote this paper about basically arguing that these particular kind of conspiracy theories were related to people's rejection of science. It turned out that they had regressed out age and basically claimed that this particular relationship existed, or sorry, that a particular relationship was not there. So they claimed that age turned out not to correlate with any of the indicator variables. There was, and they had said in their methods, they ran people from like 18 to 40 or something. There were two age outliers, 5 and somebody who was 32,757 years old, right? Which is clearly just some sort of weird integer mix-up. And it turns out that when you do it properly, there's significant relationships there. So they had to basically correct the paper. Obviously, all they would have had to do is put in one statement that basically says, we know that the ages should, for every variable, where we know that it should be within a particular range, we should put in a test to make sure that the data are actually within that range. Reaction times should not be longer than however long you give people to do the task, right? Ages should not be longer than the oldest human, or longer than whatever your age range is supposed to be in the study. So finally, we've already talked a lot about making your data, code, and results public, and this comes back to the comment that you made, right? That so Nick, what's his name? Nick Barnes had a paper basically arguing the opposite of what you're arguing. That if you have code, you should put it out there regardless of how good, how well commented, or how crappy it is. And I think there's arguments to be made in both directions, but I think we have to be careful not to sort of make people scared to put out their code because they're going to be seen as bad programmers or seen as not being useful. I'll just mention another concern people often have about putting out code is, oh, I'm going to have to support it, right? People are going to be emailing me with problems. And that's that does happen. I still get emails about the SPM ROI toolbox that I'd written like 15 years ago that I deprecated. I shut down the source for it page like 10 years ago. But one thing that can help with that is something that we've done for some of our code projects is using sort of open forums like NeuroStars or something like that, where basically you can have, instead of people emailing you and you sending them back answers, they ask the questions in a public forum. So then the answers are in the public forum as well, so other people can come and look and find those answers. So I think that's a useful thing to think about. So I'm going to end with this slide because it has my favorite Richard Feynman quote, and then I'll take any more questions, and then we'll also do an open neuro demo. it's way too easy to fool ourselves, and a lot of the mechanisms of science kind of push us towards fooling ourselves and not away from fooling ourselves. And so we're trying to think about how we can actually, redesign the way we do science. And obviously some of this gets to these social issues that are a lot harder for someone like me to redesign. Working with, really good software people, we can at least redesign the technical stuff. And hopefully that will help us start to do a better job of not fooling ourselves. And particularly want to thank Chris. Oscar's not here, but Chris and Oscar, who've done a ton of the work on the software development side, and also thank Tal, who we've collaborated with for a number of years on some of these projects. So let me first just ask for questions, and then I will bring up Open Neuro. And hopefully not crash. Questions? Yeah.
So all of these things take a lot of effort. How do these feature into grants, and how open have been granting agencies to these kind of initiatives within the bigger picture of the problem?
Yeah. So one thing to say is that at least in the U.S., the grant agencies are highly concerned about reproducibility. I mean, NIH just added quote unquote rigor and reproducibility criteria to the grant review. They don't always get used in the right way by the grant reviewers, but nonetheless, they're at least signaling that this is a really important thing. And I think reviewers are starting to, it's certainly like I'm on a NIH review panel, have been for 5 years now, and these issues are becoming more and more, in part because I bring them up more and more, but others on the panel also, I think, are increasingly concerned about things like sample sizes and data sharing and stuff like that. So I think it's a slow process, but those agencies are really are really trying to push it. I think like NSF, you now have to have a data sharing plan with any grant you send in. And if you're not going to share data, you have to say why that is. So I think it's moving in the right direction on the grant side. I don't know how it is in other parts of the world, like in Europe. But yeah.
Are there any publications that you recommend for having code reviews that's like kind of part of their service?
That's a good question. I don't know of anything offhand. Do either of you know about a good paper that lays out kind of like best practices for code review? I mean, I know there's.
This is a good blog post.
Oh, okay. Can you Slack that?
I'll Slack
cool.
That. It's short, but that's kind of what we want to project.
Yeah. Cool. Okay.
I think Greg Wilson would probably have the best answer for that from software carpentry. But I don't know what that answer is.
Huh.
There's a book called Code Smells.
Code smell?
Yeah, about finding different aspects of the code that looks fishy.
Interesting. Huh.
I agree with the code smells. There's several good software engineering books out there, but I also want to caution you because we're not working for a company, and many of those books were designed for working engineers.
Yep.
And they're working for a large corporation, which needs a lot of best practices. And well, their best practices are for their practice, which is not our practice.
Mm-hmm.
Maybe that's a project pitch then.
Yeah. There's something different about research code inherently in its purpose and its design, which won't be the same as the best practices of, I don't know, whatever.
Yep. No, I totally agree. Yeah. In fact, Oscar and our group is working on one of these 10 simple rules papers now around sort of software testing, and it's very clear there that the things that you do in industry are not necessarily the things you want to do, and some of them are, but some of them aren't. Yeah. Sorry, there were other questions. Yeah, Dave.
Russ, this was great. I really appreciate you rounding everything up for us here. I wanted to push a little more on the grants stuff. If you double your sample size, costs twice as much, it's going to look like you're doing less science, potentially, that somebody comes in, maybe just naively, and proposes twice the number of projects, and it's going to get a lot more done. So in a study section, what's that look like?
In general, I mean, my study section, in part because I think I've been talking about it long enough that others have started to buy into it. We generally, people would get penalized for having too small of a sample. They would never get penalized as long as the larger sample is motivated by a power analysis. We would, in general, I think, that would give them bonus points, right? Having a realistic power analysis and having, a sample size that we all feel like is reasonable, whereas, people who have a clearly bogus power analysis that tells us they only need 8 subjects to find an effect, they get beaten on badly. Okay, so let me just show you what OpenNeuro looks like. So this is a project that we've been developing for a couple years now, which is meant to be sort of the next generation of OpenfMRI. So OpenfMRI was a place where somebody would want to share their data. They send it to us, or we help them get into BIDS format, and then we put it on the web. OpenNeuro, so part of the problem is we need to incentivize people to share their data, and just saying, hey, do a bunch of work to get it in BIDS format and then we'll share it for you, doesn't really give them much except for like a link to put in their paper if the journal requires data sharing. So we thought about, what can we do to get people to incentivize them to share their data? And one thing we thought was, well, we can give them stuff in return, right? The idea is kind of like NeuroVault, where NeuroVault, you share your data, you get all these toys you can apply to the data. Here, we also want to give people tools, and particularly want to give them free computing. So the idea is, you have a data set and you want to run FreeSurfer on your 100 subjects, you could install FreeSurfer on your cluster and go do it and wait for a while and hope that it worked right. Or you could upload your data to OpenNeuro and punch a button and come back some number of hours later and all of your 100 subjects have been analyzed using FreeSurfer, and you could download the results and it didn't cost you anything. And you know exactly what version of FreeSurfer was used, and if you want to share the results with one of your colleagues privately, you can do that, or if you want to share them publicly, you can do that. So that's kind of the vision. So let me just show you kind of what the experience looks like. So you sign in with your Google account. So I'll just sign in. Anybody can sign in. You can also browse publicly, so there's certain data sets that are available publicly. I'm going to show you what it looks like, once you have actually signed in. So I have a couple of example data sets here, but let's say that I wanted to upload another data set. So the idea is I hit upload data set. It asks me for a BIDS data set, and I'm going to go find one. So this is just one subject's worth of data, and we'll just call this BIDS test. And so what it just did was looked at the data and validated that it's proper BIDS format. It took, like a second or something. And so made sure that it's in proper BIDS format. If it wasn't, it would come back and give me an error. And then I have to agree that, one, that I've de-identified the data, and two, that if you do an analysis on the data on more than one subject, you're basically agreeing that the data will be made publicly available within no more than 18 months. So if you basically don't click the Make Public button, we will click it for you in 18 months. I'm going to cancel that because I already have this data set. But then it would go basically upload the data set. If it gets interrupted, it can sort of reload. So it's pretty smart about uploading. So the idea is that you have to do the work of putting the data set into BIDS. We kind of help you in showing you, oh, hey, there's a piece of metadata that you need. You haven't told me what the repetition time is, or you haven't told me what the task name is for this particular task. But otherwise, it'll just let you bring the data in. So I've got a couple data sets here. So let's go look at the MyConnectome example. So when I upload the data set, it's sitting in what's called a draft version right here. Now, when we perform analyses on a data set, we want to know exactly what was in that data set. So we don't let you perform analysis until you've taken what we call a snapshot. So the idea is you have this draft data set, and I could, if I hadn't already done it, I could click Create Snapshot, and it would go create this. This is the version 1 snapshot. And you can see this doesn't have much. It just has one subject, one session, MyConnectome data. And it doesn't have much in terms of the information down here, but if you had put all of this into your BIDS data set, it would know how to acknowledge the paper. In fact, I can go out here and show you an example of one from the public data set. So we have a bunch of the OpenfMRI data sets in here now. And so let's go, for example, to this one that was uploaded by a cheeky person who calls himself William James. And this tells you, it's got a README file that has a bunch of information about what was done. It has the license, it has acknowledgments, has how to acknowledge the paper. So please cite the papers listed in references and links, has information about the funding, and it has all the papers. So it basically would say, if you're going to use these data, please reference those papers. So hopefully it's a way to help people get more visibility of their papers. So let's go back to my data set. and we can look at that snapshot. And so once we have a snapshot, now we can go do some analyses. So I click that button which says Run Analysis, and it's going to basically say which snapshot do you want to use, and then it gives me a bunch of things I can do with this data set. So it's looked at the data set to figure out what types of pipelines can be applied to this data set, and then will let me, so let's say I want to run the HCP pipelines on my data set. Now it might okay, so it actually is fine, so I could do that, and then I'd have to put in my free server license key, and if I wanted to, if I just had one subject I wanted to run, I could tell it which one, or I could just tell it to run all of them, and I say start, and so now the analysis has been submitted, and you see over to the side here we have our panel of analyses. So you can, if you look at the HCP pipelines, it's running version 5, it's currently uploading the data. We can also go look at something I ran earlier. So remember I mentioned fMRI prep, which is this pre-processing workflow. I ran a version of this. It actually succeeded. And I could see like what were the parameters that I set. But more importantly, I can look at the results. So I can, for example, go into fMRI prep. I could just download it all as one big thing if you didn't want to look at it online. But I can also go, for example, let's go look at the report that fMRI prep spits out, right, which is going to tell us, a bunch of stuff about what was done, what the data looked like, and then it's going to show us, things like the brain mask. Where is the brain mask? Oh, yeah, my color blindness is messing me up. So you can see the little red line. You can see the segmentation. You can see the T1 image over the anatomical image, so you can see it kind of morphing between. It's kind of hard to see because the registration actually worked really well, right? But you can kind of see it morphing, so a few things change. So this is actually a great way to see how well your subject, each of the subjects, is aligned with the structural. And then you can see an example of the surface reconstruction, and then get a bunch of information, for example, about how well the EPI to T1 transformation went. You can see we do all these things to extract nuisance variables, like to extract, so there's a set of tools called CompCore that try to basically pull out signals that are sort of junk, like respiration and things like that, from like high variance voxels. So you can see these are the voxels where we think the junk is coming from, and then if there's errors, they'll give you errors. So basically this is the glass box idea, right? We're basically giving you a report for each of the different things that happens, and you can look at them on the website, or you can download them. One of the cool things that Chris and Oscar have done is that these HTML pages actually have all of the images embedded in them, so you can just send somebody that, if you want to email it, you can email that page, and it might be a big file because it has, images in it, but you don't have to worry about like links within the file or anything. It's a self-contained HTML file with all of those images in it, which is sort of handy for sharing. Yeah? So
this is for one subject. Does it create it for each subject when you run multiple subjects?
Yeah, so let me go back to, I don't know that I have fMRI prep on multiple subjects, but I have Mri Qc, which does similar sorts of stuff, so I can show you what that looks like. So this is the T1 weighted imaging from, I hope I had Mri Qc, do I? Let's see. It might still be running, let's see. Succeeded, okay, good. The website is still at a point where when things succeed, we're like really happy. So let's go to, so I'm looking at the Mri Qc reports. This is a data set with, it says 14 subjects. It's really 14 different sessions from me. And so we can go into the reports here, and where's the group, T1 weighted group report. I can view that. And so it does actually these cool things where it shows you all of the different subjects, like on there. So let's say for one of these measures like SNR, right? So one of the sessions has like much higher SNR in the gray matter than the others. So if I want to dig into what's going on there, I just click on that subject, and then it brings up their individual report, and then I can go look at just visually what does it look like. And I can look at the like standard deviation map. And I don't know, yeah, it's sort of coming up. So basically you can go look at some of the QA. If it was functional, you could go look at motion and stuff like that.
For fMRI prep, pipeline that
No, not right now. There's a little bit of customization. Actually, Chris could speak more about kind of where it stands currently.
Well, there's some level of flexibility. You can choose, for example, not to do surface if you don't need to do surface, or not to do normalization if you want to do using stuff with analysis. But we are really striving to make it robust in a way that that you wouldn't have to tweak those things like like what particular starting points of co-registration and things like that. It's supposed to work out really well. So those things.
And we're in the process of testing it across like all the open fMRI data sets to basically see where it fails, are there particular types of data it fails on, to try to sort of make it more robust to those. But the idea would be any data set you give it, it should be able to do the right thing with. Which obviously, there's always going to be something that breaks it, but we're trying to get as far out in the tail as we can. Yeah, Chris?
I guess I'm wondering, it seems like this is really useful to the extent that everyone's processing paths are parallel to one another.
Yep.
And at some point, though, everyone's going to start forking off, and now they want to do their one bespoke thing for their specific question.
Yep.
How do you see this kind of fitting into that pipeline?
I think it's, ultimately, people are always going to probably have to download stuff from here and then take it and go the last mile. It sort of depends. If you're just doing an activation study, let's say you want to know about how psychotic individuals differ from healthy individuals in cortical thickness or in activation, that is something you should be able to do soup to nuts on this platform, right? So then you'll be able to say, here are all the containers I used and all the snapshots I used to know exactly what data went into it. So it's completely reproducible. I agree that there's always going to be things that, interesting analyses people want to try. And part of the hope is that at least they'll, there's a couple things, right? One is, if you put in your data just to get fMRI prep run and FreeSurfer run and whatever else you need run, and then you go download the processed data, you've already bought into sharing, right? So the data are going to be shared at some point on the public dashboard here. And also that we've gotten you to be reproducible up to some degree at least, and hopefully it'll convince people, hey, maybe I should write a containerized version of the bespoke thing that I'm doing that I can then go share with people. Now we're pretty heavily, as you saw, there's not that many workflows one can run on these data right now. It's pretty heavily curated, in part for kind of like security reasons, right? We don't want somebody building something that goes and does Bitcoin mining in the background. But the hope is that those, as the number of bids apps grows, in principle, any bids app should be implementable on here. So, with little or more work to kind of deal with our particular Amazon setup, but that's the idea. So I think it'll grow, but there's we realize that people are always going to have to sort of take things down at some point probably to work with them. Yeah.
Is it possible to have a script of the commands you do to execute one workflow? Like what if we don't want to do it here, or there's some data set which we can't upload here but want to do the same thing as we did here for the data set which
Yeah, so you can basically, because these are, everything we're doing here is one of those bids apps, you can run those just as easily on your own machine as you can on here. And you could reproduce things exactly.
So how we get that, with what settings would they run here? Or is it
Sorry, I don't quite understand.
For example, sometimes you have some parameters for any function which you are running. So how would we like to reproduce the exact same? How do we get it?
So so those are exposed. Let me just go. I don't know if there's any interesting examples here, but those are basically there's a parameters bit where it'll basically tell you any parameters that were set. So there's no interesting parameters here, but if there were parameters you were setting, they would be sort of grabbable there. Yeah.
So I think to answer your question, it's also the report would show you one step, right? So if you run it on one data set, get the report, you basically freedom.
Yeah, I don't know that it gives you that in. It depends on what your goal is, right? Maybe Chris has more to say.
So reports are specific to fMRI prep. So this is just one app, and recently actually added this that the reports have the whole command line and what version was it and all sorts of things, lots of problems there. But in principle, bits apps do not kind of sort of force a unified way of working. But the platform itself will show all of the parameters you pass the particular app here. So you can take the same container, pass the same data set, and pass the same parameters, and you should get the same result.
Satran?
So going back to your 4 principles, one of them was to move the human in the loop, I think.
Right.
So how do you reconcile having to look at these things, especially in the context of these large public data sets, the computational variability that comes with running different kinds of workflows, and having to inspect things? What do you see as the next future steps of
deep learning for QA? No, I mean, I think you're right. That's the place where clearly humans still have to be in the loop, is making sure that the stupid computer actually did the right thing. And we don't have, Google hasn't built us something that can do that computationally yet. So I agree that you would like that. I mean, it depends on, humans are really good at seeing things that computers aren't unless they have enough data, right? And so it's probably never going to be the case that we're going to be able to build something that is going to catch all the things that a skilled, experienced human is going to be able to find. So we're probably always going to need to be in the loop. But the particular place where I don't want us to be in the loop is where you're doing things that could directly change the analysis outcomes, right? I mean, in a sense, you could here by like deciding that I'm going to throw out particular subjects, right? But you would hope that you have criteria that drive you to do that. Whereas, like if you're circling the amygdala by hand and, you just happen to know that like this subject is a PTSD subject and my hypothesis is that the amygdala is larger, and so I might, those are the kind of things that really worry me, where the human is like directly in the loop, and if the human has knowledge, isn't perfectly blinded, then they could actually bias the results. Or just, doesn't happen to check that they pulled the box far enough down on Excel.
Okay, I think in the interest of getting lunch, we'll stop here. Thanks, Russ.
Yep. Thank you. Can I just add? I'm going to just add one more thing, which is, obviously, I hope you'll all come on and play around. If you find bugs, please don't just go away and say their platform is crappy, but tell us what the bug was. You can still go away and say it's crappy, but tell us what the bugs were, what the problems were you found, so we can try to fix them.
And what we're going to do is we're going to do lunch over back at Blender Hall, and then meet where we entered this building at 5 minutes to 1, which is